Hi, everybody. Hopefully you're doing well. My name is Daniel, and I'll be giving you a presentation on reproduction and development for Bio Class 12. So a little thing about us, we're a nonprofit in the Commonwealth of Virginia. We can help you out through any Slack email questions you send us. This session is going to be recorded, so you can view it at any time, which hopefully you're doing now. And yeah, our email is right there, info@premedpeers.org. Any questions you might have. Yeah, so feel free to reach out to us. So a little bit of an outline is we're going to be doing a little bit of review. Then we'll do a couple warm-up questions, content review, which will be the main focus of this session. And finally, we'll do a practice passage, which will reinforce the skills we've learned in this lesson. So just a little overview. First, we'll be talking about the male system. So spermatogenesis, the pathway of sperm, gender development, sexual function, and then we'll talk a little bit about the female body, external genitalia, the ovaries, oogenesis, menstrual cycle, stem cells, fertilization, fetal stages, all of that stuff. So first, a warm-up question. What is a totipotent cell? So a couple options we have here. And it's a cell that has the ability to form a complete organism through embryogenesis. So for this question, we'll be talking a little bit about pluripotent, totipotent, all the different types of cells that you have as stem cells. But basically that from these options, the best choice is choice B. Most of these, they're just kind of stating a fact or something that's not really relevant to the question. And a cell that has the ability to differentiate into any type of tissue, if exposed to appropriate chemical signals. We didn't really know anything about chemical signals yet. We're just talking about what is a totipotent cell. So, excuse me, an environmental toxin has been found to directly disrupt normal gamete function production in women. What tissue or organ is the toxin likely localizing to? So basically, this requires knowledge of female reproductive anatomy, and the answer is ovaries. So for the oviduct, that's also known as the fallopian tubes. That's where fertilization tends to take place, and that's between the ovaries and the uterus. The endometrium, that's the lining of the uterus, and it's basically where the embryo will eventually implant and start to grow and gain nutrients that way. And then lastly, the uterus is basically the outside of the endometrium. So the ovaries, they're the equivalent, the female version of testes. Those are gametes. Those produce, different hormones. They are involved in sexual function. Which of the following describes the function of follicle-stimulating hormone in females? So basically, this just requires knowledge of hormones and what they do. So it's choice D. So basically, milk production, you think sometimes oxytocin, but most of the time you think prolactin. And increased basal metabolic rate, those are thyroid hormones, T3, T4. Stimulate estrogen secretion from ovaries, that's LH, luteinizing hormone. We'll talk a little bit about that. Basically, follicle-stimulated hormone and luteinizing hormone, they work together, but they have slightly different end goals. And lastly, promote growth of immature follicles in ovaries. So this is the correct answer. Even if you really didn't have much background knowledge, a good guess would think follicle stimulating, okay, you stimulate in some sort of way, maybe you're stimulating the growth of the follicles. So now we're going to get into some content, and we'll first be talking about the male system, focusing on testes and spermatogenesis. So this is a good overview of the topic at hand from kind of like a macro and micro perspective. So basically, you're looking at the inside of the scrotum right now, where the testes are. We have the epididymis. That's this part right here. And basically, inside the testes, there are just a lot of complex tubes, many tubes all around, kind of like little noodles, I guess you could say. And within these tubes, there's a lot of growth and nurturing going on for sperm cells. And ultimately, these develop mature sperm from a germ cell. So Sertoli cells, they kind of are like aiding the process, helping them mature. So a little bit of anatomy is that the testes or the testicles or male gonads, they lie right behind the penis. And the pouch of skin is called the scrotum. So it's somewhat muscular, and it's basically its function is to maintain heat. So your testes are very sensitive. So if it's very hot, they're gonna kind of fall a little down away from the body, so that the heat tends to be lowered. And if it's very cold, I believe it's called the cremastidor muscle. It's going to pull them up tighter, so it's closer to the body's heat. So it's very important to know the pathway of sperm as they mature. A good mnemonic is 7-up. And this is the seminiferous tubule, epididymis, vas deferens, ejaculatory duct, nothing, and then the urethra, and finally out through the penis. So it's a little confusing. The N here, it stands for nothing. So that's kind of, you don't think about that. But yeah, here's basically looking at the anatomy. So we have the tubules right here. So as you can see, this is where they're first starting to mature. There's a lot of them. There's a ton of them. And even each section has a ton of tubules inside of it. So that's where they ultimately start. Then they follow their way up to the head of the epididymis. So that's basically kind of like a little half covering of the testes. They go down, they go up to the vas deferens. And that's right here. It kind of goes around, kind of around the bladder. It doesn't go in the bladder at all, but around the bladder. And finally, it goes up here through the urethra, and then lastly out the penis. So just some overview between diploid and haploid cells, because this stuff gets a little confusing. So just talking about the basic genetics. Diploid, you're thinking of 2 copies, 2N. So here we have 3 pairs, but they're still diploid because there's 2 of them. They're pairs. Versus haploid, there's 1 copy of each chromosome. So here we have still the same amount of chromosomes, but they're haploid because there's only 1 pair. So this is important in meiosis, which is production of gametes. So that's what we're going to learn. So in spermatogenesis, it's the process in which sperm are formed in the tubules of the testes, like I said. So diploid spermatogoum, they undergo mitosis 1 to form diploid primary spermatocytes. So we're still diploid, we're still 2N. And then next, 2 haploid secondary spermatocytes are formed, and these undergo meiosis 2. And then this end product is 4 haploid spermatids. And then these undergo further maturation, and they finally develop into spermazoa. So here's a little chart of it. So we have diploid, diploid, haploid, haploid, haploid. And as you can see, they're kind of just more like cells. But then finally, when they develop into spermazoa, they finally have their tail and their head, and they actually look like sperm. But yeah, this process starts at the beginning of puberty. So at birth, this process isn't happening, and then it continually happens. And this is very high yield stuff. This appeared on my test. A good mnemonic is go catch the zebra, because, spermatogoum, spermatocyte, spermatids, spermatozoa, there's not that much difference between them. So you got to go for the first letter. That's a little different. So this is just the order and maturation. So the spermatogoum, they're very immature, and then the spermatozoa, they're completely mature sperm cells. So some of the sort of the 2 helper cells of this process you'll need to know are the Sertoli versus the Leydig cells. So like I said, this meiotic division and maturation of sperm occurs in these very long, coiled, wrapped around structures in the testes, the tubules, and it's aided by these 2 cells. So the Sertoli cells, both of these are specialized cells, but they're stimulated by a follicle-stimulating hormone. And then the Leydig cells, they're basically in the same area too, and they also help as well, but they're stimulated by a luteinizing hormone to produce testosterone. So think Leydig cells, think luteinizing, so the two L's, and also just think of testosterone. So all of these processes are interconnected, but think Leydig cells stimulated by a luteinizing hormone produce testosterone. The Sertoli cells, they basically just help with the maturation, and they're stimulated by follicle stimulating hormone. So now we're going to talk about the sperm pathway, and we talked a little bit about this with the 7-Up mnemonic. So basically, once they acquire mobility, which is when they get their tail, they can finally move, they're completely mature. They wait until they're ejaculated out in a structure called the vas deferens, and that's right here. It's just basically a long tube from the epididymis to all around into the penis or urethra. And basically, when ejaculation occurs, the sperm will pass through this tube, through the ejaculatory duct, the urethra, and then finally it'll exit out through the penis. So a little bit about the anatomy. The acrosome, that's kind of derived from the Golgi apparatus, that organelle that you probably learned about. And basically, you can think about it as a cap. Basically, it's got a ton of enzymes in there, and it allows it to break the zona peculia. Basically, that's the outer covering out of the egg or the oocyte. Yeah, so it allows it to penetrate through that during fertilization. And this is a very important process because you don't want any, let's say, germs or immune cells to attack this egg because it's very valuable. But at the same point, you don't want a ton of sperm to attack the egg, because if you have a bunch of sperm in one egg, it's just not going to work out. There's going to be too much genetic material in there, and you're going to have genetic abnormalities. So that's a very important process. Basically, the nucleus, that's obviously, it houses the DNA. In this case, like all cells, it houses the DNA, but it has a very special property in that it's a gamete. So that genetic material is basically the entire point of the sperm. The mid piece that houses a ton of mitochondria. So as you know, mitochondria are responsible for energy. So basically, there's just a ton of ATP in the mid piece, and the mitochondria use this to power the tail or the flagellum. Basically, that's what provides it the motility to travel through the fallopian tubes and eventually find the egg or the oocyte. So next we'll be talking about the female system. Just pretty basic overview of the external and I guess internal genitalia. Basically, so you have the vagina, the outer area, the uterus. That's where the egg will implant and start to develop. The cervix is basically kind of the intersection point between the two. The ovaries, we learned those are the female gametes. They're responsible for hormone production, follicle stimulation. And then lastly, the fallopian tubes, those are the tubes that travel between the uterus and the ovaries. So oogenesis, it's a process that's very similar, but also has a lot of differences. As you can see, it's a it has mitosis initially, but basically when you produce the gametes, it's meiosis. So basically we have the diploid cells, and then ultimately they become haploid. But it's very important to know that at birth, all of these eggs start to develop, but as you can see, right after, well, not right after, but at prophase 1 in meiosis, they're arrested. So that just means they're stopped until further development. So, and also it's very important to know that unlike sperm, basically every sort of additional process, it's not dividing, well, it is dividing, but ultimately you're going to develop a polar body. And a polar body is basically, you can think of it as the exact same thing, but with a lot less cytoplasm. So a question you might be asked is the genetic material in a polar body and an oocyte, is that the same? And the answer is yes, it's actually exactly the same. They're exactly the same cells. The only difference is that the polar body has a lot less cytoplasm. So as a result, it's a lot smaller. It can't function, and it basically just undergoes atrophy and dies. So in the end, you only get one ovium. So basically, the ovaries house ova or eggs, and they also are responsible for producing estrogen and progesterone. And oogenesis is the process in which these eggs are produced in females. Spermatogenesis, it begins around the onset of puberty, like I mentioned, but the difference is oogenesis begins before birth. So even before you're born, this process is happening. And basically, diploid ovarian stem cells, they undergo mitosis to form diploid primary oocytes. Um, as we see the primary oocytes, and this is what's halted. Yeah, so they're halted at prophase 1 until puberty. Once puberty happens, then they're allowed to proceed. So excuse me. Once puberty happens, mitotic fission of these oocytes will continue, and basically during each menstrual cycle, excuse me, um, one of these primary oocytes is selected. Again, all of these, they're halted from birth in prophase 1. So one of them is selected, and this selection process happens basically due to hormones. Basically, all of them start to grow, and basically whichever egg is the largest, basically will kind of keep on growing, and all of the others will kind of just shrink a little bit. Um, So it's going to finish meiosis 1 and produce a haploid secondary oocyte and a much smaller polar body. So again, these are just discarded. They're ignored. And then the secondary oocyte initiates meiosis 1, but it stops at metaphase 2. During ovulation, the follicle that the oocyte is encased in will rupture, and this happens around day 14. Day 14. And that developing oocyte it's released out of the ovaries, and it eventually enters the fallopian tube where it may be fertilized. So basically, it needs a sperm cell to continue along its process, or else it's kind of trapped in metaphase 1. Metaphase 2, sorry. So these eggs that do not complete meiosis 1 until a sperm binds to the oocyte, and then once binding, it'll finish meiosis 2 and divide into 2 cells. Again, there's a larger ovium, which basically has enough cytoplasm, and then again, there's another second polar body, so right here. So basically, you need a sperm cell and fertilization right here for this to continue to get the second polar body. And then the zona pelluca, I'm probably mispronouncing it, but that's basically a thick outer covering that protects the oocyte, as you can see here. And it's important because, like I mentioned, you want to make sure that specifically sperm cells enter through this egg, and specifically, ideally, you only want one sperm cell, right? Maybe if you have a couple more, it's fine, but if you have a ton, that's just not going to work out. So next, we'll talk a little bit about the menstrual cycle. So basically, you can divide it into 2 parts: the follicular phase and the luteal phase. Again, I'm probably mispronouncing them. But basically, there's the endometrial cycle, and then basically the hormone levels. So as you know, this is the lining to the uterus, and according to whatever hormones are present, basically it's either building up or it's destroying itself and kind of leaking out in a period. So basically, the cycle is around 28 days, but that's just average. It can be longer, it can be less. And around midway through, that's when ovulation happens. That's when the oocyte is released from the follicle, and it begins its journey down the fallopian tube. And then, so it's developing, and then after it's released, then the follicle its work is done, so it kind of starts to just shrivel up and die. And at first, it's called corpus luteum, sorry, and then a corpus albicans. So basically, that's just like the first step, then the second step, and then eventually it just atrophies and dies away. So basically, there's a very complex interaction between these hormones, but basically, what you need to focus on is an important thing to know is the luteinizing hormone right here. This is called luteinizing surge. So it immediately precedes ovulation. So that's probably the most distinct feature about this chart. What precedes ovulation around day 14? You want to know that's a huge surge in luteinizing hormone. Then also you should also know that estrogen, a useful mnemonic you can think about is estrogen established and progesterone protects, and in relation to the endometrium. Because you can see estrogen peaks around right before ovulation and then it dips down, whereas progesterone slowly increases as the luteal phase goes. So basically estrogen established this endometrium, which has been shedded out, and it slowly grows, and then eventually it sort of reaches a peak where progesterone protects it. And this is so that right at this point around day 14, the egg is traveling along the fallopian tubes, and hopefully, depending on your situation, it gets fertilized, and then it goes down into the uterus. And the idea is that it's very mucousy in the endometrium, so it's going to implant itself against the walls. And you want to have a very lush vascular endometrium. If an egg, for whatever reason, landed in the endometrium around this area, it probably would not survive just because the mucus might not be sticky enough and there's just not going to be enough nutrients. So basically, that's what this whole process is. That's what its goal is. So just a little bit about fertilization and cleavage. I'll just be touching a little bit about it. There's a lot more to it, but basically, so once this egg is fertilized, it undergoes a process of cleavage. And cleavage, like you think of, it's cutting, it's separating. The important thing to note is it's not growing. It's actually still remaining the same size. It's just getting cut down. So it's kind of like these two cells are half the size of this egg. These four cells are a quarter of it, right all the way to the 16. And these are just names for it. First two cell, obviously up to the 16 stage. Once it gets past the 16 stage, they kind of give up naming it by that, and it gets another name, morula. And then finally, the cells start to finally differentiate and proliferate a lot more. So blastula, and then finally it starts to separate into it starts to differentiate into different functions, into the three germinators specifically. And at this point, it's called a gastrula. A good mnemonic for this is must be good. So MBG, MBG. So yeah, it develops into the three germ layers, which we'll be talking a little bit about later. So one of the warm-up questions was talking about the stem cells. So basically, initially, when you have these cells, obviously it starts from one cell. So you got to imagine, how does all these different cells in the body with very different and unique functions, how do they just develop from one cell? Basically, so this goes into that. The totipotent, those cells, think like total, think everything, or like totalitarian dictatorship. They can be anything. Excuse me. They can be any form of cell. The pluripotent, they can make any cell of the organism, not the placenta. So that's very important. The only difference between these two is that these can become the placenta if they want to, depending on the signals and different chemicals. These can be anything in your body except the placenta. And then lastly, the most differentiated out of all of these are the multipotent cells. So these can make multiple cells. And basically, by that meaning is that, like, you can kind of think of like a group of cells, let's say like nerve cells or muscle cells. So in theory, a multipotent cell could be like, you know, the muscle in your bicep, or maybe even the muscle in your heart, or maybe even smooth muscle in your digestive system. But a multipotent muscle cell couldn't somehow become like a neuron cell or something like that. But yeah, so for example, like a blood cell could be like white blood cell, red blood cell, but it's not going to be turn into a muscle cell. So yeah, so think like totalitarian, whole, so it can be really anything. It could be, and then the pluripotent, it's basically focusing, on the inner cell mass, and then multipotent, that's like a set of cells. So in this example, it could be different nerve cells, but it can't be red blood cells or adipose tissue or anything else. Lastly, we'll be talking a little bit about embryogenesis. So these are now the three germ layers that I was talking about: the ectoderm, the mesoderm, and the endoderm. These are very important. They'll definitely show up. So the simple way to differentiate these two at face value would be ectoderm, think outside, mesoderm, inside, and endoderm, like really deep inside. So basically, the ectoderm, think skin, obviously, eyes, mouth, any orifices like your anus, really anything along those lines, any linings. The mesoderm, that's basically everything else. So bones, muscle, internal organs, the gonads, could be like the heart or something like that. It's very important. It's a little more low yield, but you might get questions that really go deep into this. So for some questions might ask you, what germ layer is the dermis? And this is a tough question because you think dermis, you think skin, but actually it's the outer layer of the dermis that's considered like your skin, right? The inner layer of the dermis, that's actually somewhat vascular. It has somewhat fat and things like that. So that's actually part of the mesoderm. I just included that because you should be aware that you might get more difficult questions that really go deep into that, unfortunately. And then lastly, the endoderm, that's like basically internal tubing. You can think about that. So like the digestive system and respiratory tracts. So you can think about that as like, that's almost like the outside inside of you, right? Like kind of weird to think about it, but if you think about your mouth through your anus, that's just like a tube to the outside. So that's what the endoderm is. And then also how they're formulated, basically the order after a zygote develops. Basically first it's the ectoderm, then the endoderm, and then followed by the mesoderm. So you might get a question like, oh, which develops first, bones or digestive system? So you not only need to know what germ layer they are, but also the order of the development. So lastly, we'll be going through practice questions. And also I was trying to find a passage on these questions, but I looked through Khan Academy and all sorts of resources. I could only get discrete questions, so I apologize for that. So a radioactive marker was applied to the mesoderm of a mammalian embryo. Which tissues or organs would have marked in this adult? So basically, this is just a very simple, discrete question. Which of these is the mesoderm? So obviously bones and muscles, that's the mesoderm. The brain, not really. The spine's a little confusing because they could be talking about the bones in the spine, which I think they are, or they could be talking about the nerves in the spine. But either way, the brain is part of the ectoderm. Skin, that's also not that clear because you could be talking about the dermis or the hypodermis. But basically, intestines are also the endoderm. And then muscles and skin. Skin is probably the ectoderm, but muscles are definitely the mesoderm. So the answer is probably A, bones and muscle. Basically, so the lungs, spine, and muscles are derived from 3 different germ layers, which would be the correct order. So basically, this is a difficult question because it not only asks you which germ layer these 3 things are, but also what's the order. And as I mentioned, we're going to have the spine first, then the lungs, and then the muscles. So the muscle is the mesoderm, the lungs, the endoderm, and the spine. I guess in this case, they're considering the spine to be the nerves in the spine. So that would be the ectoderm. Follicle stimulating hormone stimulates the growth of a follicle in the ovary, a process. So basically, this is just, you don't even need to know about follicle stimulating hormone to basically know when we're talking about stimulating growth of follicles, we're thinking about menstruation, we're thinking about reproduction. So it's, you don't want to get confused because the eggs are arrested in prophase before you're born. That's correct, but we're not growing anything after that until puberty, and then menopause. That's when periods stop. Basically, when you get to a certain age. So that would be the answer. Basically, whenever someone is going through their period and their menstrual cycle, that's when these follicles are going to be stimulated.